McFadden is professor of molecular genetics at the University of Surrey. He has worked on human genetic diseases, infectious diseases, and has researched the genetics of microbes. He has published numerous scientific articles on subjects as wide-ranging as bacterial genetics, tuberculosis, idiopathic diseases, and computer modeling of evolution. He has contributed to many books and is also the author of a successful popular book of science, the last one being about the coming of age of quantum biology. It is precisely about quantum biology that Professor McFadden will tell us about in his talk today.
Thank you.
Thank you.
Good morning, everyone. And first, I'd like to thank the organisers for inviting me here today. It's a great pleasure to speak here. So I'm going to be talking about quantum biology. And that's really the question that as a biologist, I'm a biologist, has always intrigued me. What is life? How did it get here? What is the essential feature and characteristic of life? And why does all these stuff, why are they so different from all that kind of stuff, despite being made of pretty much the same materials? This is the central puzzle of life, and one which I don't think we've yet really answered. The famous quantum physicist Richard Feynman once said, "What I can't make, I don't understand." And no one has ever managed to make life from these kind of materials. The only way you can make life is the same messy process that started nearly 4 billion years ago. So, if we believe Feynman's motto, then we don't fully understand life yet. So, what I'm going to do is make a case for saying that life lives on the edge between the classical and quantum worlds. And you've heard quite a lot already about the quantum world, and we'll know some differences between that and the classical world. The classical world is the world of Kepler's solar system, Newton's apple falling, steam trains. And the key aspect of it is that at a macroscopic level, it's all structure and order, such as planets or steam trains. But at a microscopic level, it's chaos, random motion. The quantum world is kind of different. The quantum world is behind this random motion, but at a microscopic level, it's structure. The orbitals of atoms, etc., provide structure. So at the bottom is structure and order, and then at the top is the randomness that gives us quantum mechanics, and then at a higher level emerge the laws of thermodynamics, etc., which bring order. Now, we've already heard about the double slit experiment already a couple of times today, so I won't go into it except to very briefly remind you the difference between waves and particles as revealed by the double slit experiment. Waves can be in two places at once. A single wave can pass through two places at once and generates these kind of interference patterns on screens. Particles such as sand are either in one slit or another, so you don't get interference patterns. And really, quantum mechanics is all about the realization that fundamental particles can be both. They can be both waves and they can be particles. Okay. So, in a way, we can kind of think of the classical world, classical particles, as being like a stone dropped into a pool. That's a particle. Bang. Makes a hole in the pool. The quantum bit are the waves. And say if you detect an electron, it will form a dot on a screen, but immediately after that detection, it behaves like a wave. Our important aspect, and I want to emphasize this because it often causes confusion in the area of quantum biology, is the wave aspects of fundamental particles require this aspect you can see here, coherence. Thus the wave is coherent across whole dimensions of the pool. Quantum mechanics requires that level of coherence to demonstrate the kind of interesting quantum phenomena we've been hearing about already, particularly, say, entanglement, but a few more phenomena we'll be looking at. And the first criticism of it is it perhaps isn't likely. So these are the kind of quantum phenomena that we see, the double slit experiment, which shows coherence, that a particle can be in two places at once. That's what we would call coherence, or sometimes called superposition, that a particle is in two places at once, or two states at once. Tunneling is another interesting quantum phenomenon that allows particles to tunnel through a barrier that would be otherwise insurmountable. It allows particles to walk through walls. And then entanglement you've already heard about. But I want to emphasize again the quantum weirdness, if you like, the entanglement, the coherence, the tunneling, all of those depend on coherence, which is very, very delicate. Much more delicate than balancing a pencil on your finger. And it's easily lost in hot, complex, molecularly noisy environments. And now, here we go. So quantum coherence requires stillness. It won't work in stormy molecular waters. And this is extremely important when thinking about the limits of quantum mechanics, when we try to understand what it is involved in, such as life. Are we the calm waters of a pond, or are we the turbulent waters of a stream going over rocks? Now, this is what the membrane of a cell looks like at a molecular level. Is it stormy, likely to be stormy, or is it likely to be calm? It already is very complex. You can see all the molecules of it. But actually here, this works. It might be reading it off the stick. Okay, let's try it. No, let's try again. It is worth seeing if we do it. Let's try it from this thing. It should play.
Maybe we can play it
Straight from, yeah, let's, it's this one up here. Yeah, try that.
Yeah.
So I hate
It's a
putting
very
movies on that.
powerful processor.
Oh, here it comes. Okay. Well, I hate putting these movies on a stick. Okay, we'll have to skip it.
These are not our laptops. Were given by the university. Open with.
Try quit time, maybe.
VLC, that's the job? Okay. The third is the one. But we have to give a. You can advance me something.
Okay.
This is so we said because it's supposed to be now opening the aqua container. No. That's where it's gonna
Everyone hold their breath.
Yeah. This is going to be amazing, so this is just for you too.
Let's try.
This is suspect.
Otherwise, I'll have to dance it.
We'll see opening.
Oh, here we go. Yes.
Oh, that's worth it.
So there you go. That's it's pulled out, but that shows you what the challenge of quantum biology, and the difficulty of claiming quantum biology for big things, like, for example, brains. For quantum biology to work, all of these molecules have to be coherent. They have to vibrate in phase. That's what water molecules are doing in a pond when you drop a stone in it. For that to happen in a single cell would be pretty darn amazing. For that to happen in a brain would be of astronomically small chance. So it's important to recognize that. Okay, I think we're good to go back to the... when we come to thinking about the questions that always arise, is quantum mechanics involved in the brain? I know Max Sandos already said that his work isn't presupposing this, but we have to bear it in mind in any other claims. So anyway, but things may be different in life. This is what one of the founders of quantum mechanics, Erwin Schrödinger, said when he was considering life, and particularly heredity, many decades ago. He thought that there was a problem in heredity. It worked too well. And he claimed that for it to work this well, it needed to be based on the orderly structures of quantum mechanics. It couldn't be based on the disorder that is below thermodynamics that you saw in the earlier slides. It had to be based on structure that went all the way down. So he proposed that structure was quantum mechanics. Everything is slow on this computer. There we go. Now, in a sense, he was right, because the structure that encodes the colour of your eyes, the shape of your nose, your propensity to get one disease or another, is encoded in this molecule, DNA. And that is a quantum mechanical entity. It's made of a position of protons, electrons, etc. within a single molecule. Ask any physicist what science you need to understand where protons, electrons are within that molecule, they would say quantum mechanics. So, in a sense, he was right. Slow, slow, slow. Do I need to hit it twice every time? Okay. That's more or less what I've said, in which every atom and every group plays an individual role. This is a DNA-based pair. This is one letter of our genetic code. What I want to point out is that it's held together. The specificity of the genetic code is brought about by the position of these protons. H stands for hydrogen, a proton, an electron, but actually all that's here is the proton. So these are really three protons holding the DNA bases together. The position of those protons determines... I have to remember to hit it maybe 20 seconds before I need to move. The position of those protons determines the genetic code. There we go. Now, I'm not going to go for another movie here. There's one here, but we won't bother with it. And that should be moving as well, but never mind. At a molecular level, our life involves the manipulation of fundamental particles. Sorry? Yeah, that's the last movie, but let's not worry about it. We can live without that movie. Essentially, what happens in terms of enzymes, an enzyme's major body, is they manipulate the fundamental particles inside your cells. That's how enzymes work. This is the structure of an enzyme. This is the structure of a substrate of an enzyme, and what the enzyme does is it rips apart the molecule and puts it back together again in another configuration. That's what enzymes do, so that they are manipulators of fundamental particles. They are dealing with quantum mechanics. And that was a movie which showed it more convincingly, but it'll change. There must be some way of making this move work faster. Oh, look, it's starting to move. It was thinking about that movie for a long time. Enjoy it. Let's see if we can move it on. Here we go. So life uniquely amplifies quantum level events to the macroscopic level. So here we have quantum level events in enzymes or in DNA, and they get amplified to the level at which they make differences to people.
Sorry, Doctor, just a few seconds for the backstage.
Okay, thank you. I was hoping once I passed the movie, we might get a little bit faster. But okay, triumph of molecular biology. So since the discovery of the double helix, which was predicted by Schrödinger really, that it was a quantum mechanical entity, biologists generally have forgotten about quantum mechanics. We don't teach it to our students, and most of us know very little about it. So we've managed to get by without quantum mechanics for a long time. And the question is, do we need it? Well, now there are several candidates for quantum biology. Phenomena that are going on inside living cells that appear to require quantum mechanics, such as photosynthesis. Now, photosynthesis has made all the matter in our bodies. It's all derived from plants originally and the animals that eat plants. So ultimately it all comes from this stuff, which is the most remarkable chemical reaction in the known universe, where air and water plus sunlight makes plants. It's the most astonishing thing that we know about, really. How does that happen? We take it for granted everywhere it happens, but it's astonishing. Anyway, it does it through this complex, which is a photosystem. It's a complex of protein and chlorophyll and other chromophores, molecules that absorb light. And this is one molecule of chlorophyll. There's a magnesium atom at its center, and it has an electron that's excited by light. And that's how light is captured in that single atom. Essentially, again, a quantum mechanical phenomenon. So the first step of photosynthesis involves capture of that photon of light by a magnesium atom in the center of a chlorophyll molecule. In order for that energy to be used by the plant, the energy needs to be transported. It's transported by a process to a reaction center. Now, it was thought that that process of transport was a classical energy transfer, like energy transfer along a wire, where the energy will hop from one place to another by classical processes. But there was a problem with understanding that, and that is that the complexes are very complex. In fact, this is what they look like. So there's lots of these chlorophyll molecules that the energy can hop to, and that creates a traveling salesman problem. There are too many different paths that the exciton, as it's called, can take to get to the reaction center, which means it should be inefficient. And yet it's the most efficient energy transport process known to man, as close to 100% as we can measure. So how does it do it? Well, Greg Engel at the University of Chicago discovered about a decade ago that it uses a quantum mechanical feature, coherence. What he did was shine photons of light from a laser at a photosystem, and then he kind of looked at what light comes out again, the light echo of that stimulation. And it came out in waves, what they call quantum beats. And the only way to account for that was that the energy was traveling not by a single path, as it would do classically, but by multiple paths simultaneously. It was being in many places at once. It was behaving quantum mechanically. And that ability to travel along multiple paths at once is likely to contribute to why photosynthesis is so efficient. You know, if you could, every day you go off to work, check out all the possible routes and discover which one's going to take you there quicker, then that would make your drive to work a lot more efficient. It seems that excitons in photosystems can do that through quantum mechanics. And I think I've just said that. It's called a quantum walk, that kind of process. I won't go into that. So essentially, what we're saying is that the photon arrives as a particle, then travels through the photosystem as a wave. When it hits the reaction center, it becomes a particle again. So we can kind of consider that photon hits the reaction center, spreads out, it's converted to the exciton, and the exciton is like the wave in the pool. It travels by all routes simultaneously. Okay, so that's one aspect of quantum biology, well, probably the best demonstrated aspect of quantum biology, and an important one. It's made our bodies. But the enzymes that turn, say, glucose into DNA, protein, and the stuff of our bodies, enzymes are also puzzling. They work so well. They have efficiencies, or they can speed. They're essentially catalysts. They speed chemical reactions. And they speed chemical reactions by factors of up to 10 to the 20. Now, if you could, to get a feel for that number, if I took my walking speed and accelerated by 10 to the 20, I would have now gone to the other end of the galaxy. That is the kind of acceleration provided by a factor of 10 to the 20. So it's a huge acceleration, and it's difficult to be accounted for by the classical processes that we know go on inside enzymes. But as I pointed out already, enzymes manipulate quantum particles. And what they seem to have managed to do, remember they've been doing it for a long time, is use this feature called quantum tunneling, which allows particles to overcome barriers that classically they wouldn't be able to overcome. And essentially in quantum tunneling, a particle kind of disappears from one point in space. It's the wave aspect of it is able to travel through a barrier and pop out the other side. Can't see that very well on here, but there's something coming through when the particle hits the barrier. Okay. And so tunneling may fill the activity gap. What has been shown is that when enzymes react, they bring the substrate together, so particles like electrons and protons are close enough that they can tunnel. Now, how it's been shown by people like Judith Klinman in California and Nigel Scruton in Manchester is by replacing water with deuterium. So water is, of course, H2O. The hydrogen can be replaced with deuterium, which has two neutrons, a proton and a neutron in its nucleus, so it has twice the mass. But it behaves chemically just like hydrogen. But because it has twice the mass, it becomes much less likely to tunnel. So if you perform an enzyme reaction, and this shows one of them, it's an enzyme called aromatic amine dehydrogenase in ordinary water, and in deuterated water you should get a difference. And this difference is called the kinetic isotope effect. And if you get a big kinetic isotope effect, it indicates that the reaction is going forward, requiring a tunneling step. I won't go into the details of these as we're a bit short of time. So, in this particular enzyme, the kinetic isotope effect was around about 55, indicating that this enzyme is actually utilizing quantum tunneling to make its products. And this enzyme is an enzyme that's involved in your body. It's very similar to many other enzymes. A lot of enzymes in your body manipulate protons and electrons, move them around, and they seem to do so using quantum tunneling, another feature of the quantum world that we don't see in the classical world. Entanglement is what this meeting is about, and we've heard already a lot about how it works. The European robin. European robin is probably common, I guess, in Belgium. We have the British robin, slightly different. It doesn't migrate. The European robin does. It goes from Europe, particularly Northern Europe, down to the Mediterranean in the winter months to escape the cold and the parties. And these two ornithologists, husband and wife team, Wolfgang and Roswitha Wutschko, discovered an extraordinarily puzzling thing about the compass that they use. So birds navigate by lots of different ways, but one way is they use a compass. And the compass is able to tell them where's north and where's south, you'd think. But in fact, it's an inclination compass. It finds the nearest pole, but can't distinguish between poles. The other thing I should have mentioned on there is it also requires light. That's a bit odd for a compass. First of all, you have to think about it. How does a compass work in an animal's body? You can't imagine an ordinary magnetic compass turning this way and that, so you've got to have some kind of chemical or some kind of structure that's going to respond to an extremely weak magnetic field. How does the animal detect that? It's a big puzzle. But this was a puzzle. It's actually not, it doesn't detect. It doesn't work in the way of a Boy Scout's compass, is how I would sometimes describe them. It detects the angle of the Earth's magnetic field in relation to the surface of the Earth. Here's the magnetic field lines on the Earth. So near the equator they're nearly horizontal. Towards the poles, they're vertical, and in between, they point at an angle, and that acute angle points to the nearest pole, whether it's a north pole or a south pole. Birds' compass do that. They're an inclination compass, not a standard magnetic compass. So how does that work? Klaus Schulston in 1976 discovered that some chemical reactions were actually sensitive to magnetic fields, which was a bit of a puzzle at the time. And what he proposed and demonstrated, it was because these reactions generate chemicals called free radicals. And free radicals have electrons that are unpaired, so they are magnetically sensitive. So this allows these chemicals to sense a magnetic field. And he went on to propose that this kind of chemical reaction may be involved in the avian compass. But it wasn't until Thurston Ritz teamed up with the Wutschenkos to test this. He worked in UC Erwin in 2004. He demonstrated that the robin's compass was disrupted by high-frequency radio waves. He developed a model by which light gets into the eye of the robin and generates free radicals. And then those free radicals are entangled, and the angle at which they are held together is sensitive to a magnetic field, and it will determine the product of that reaction. The final product of that reaction will be sensitive to the magnetic field. So that's what he proposed, and it came up with a prediction. The prediction was that it should be disrupted by high-frequency radio waves. So he teamed up with the Wutschkos to test that. They put robins in a box with a magnetic field and radio frequency generator, and they showed that the birds could detect the magnetic field, the Earth's magnetic field, but not when the radio frequencies were on. So it was disrupted exactly as predicted by his theory. So that's where it is proposed. I would say the avian navigation model is still theoretical. It hasn't really been nailed. We don't know what the receptor is really for the avian navigation. There is an enzyme proposed, cryptochrome, but it hasn't really been nailed as the receptor. So there's still a lot of questions. Me and my colleague Jim Al-Khalili have been interested in DNA and whether DNA may be involved in mutation. How much time have I got?
More than 10 minutes.
Okay, I think we make this then. So, as I already pointed out, protons are the coding features of DNA. Actually, Schrödinger also suggested that mutations may be caused by quantum jumps. And the Swede, Pierre-Olaf Lodin, pointed out, he was the first to point out that DNA is essentially a quantum code written in proton position, and proposed that mutations may be caused by protons tunneling into the wrong position. So here's the canonical position of protons within the double helix. First one to point out that genetic information, as I've mentioned before, is written in proton position. And it's quantum coding that was discovered 3.8 billion years ago. So if we take away the DNA backbone, essentially the genetic code is just the position of these protons. It's a quantum code that was discovered 3.8 billion years before we learned to code with particle position at the quantum level. This is IBM written in atoms. Back in, I think, 1980s. 1989. This is what IBM did, and for the first time coded information at the quantum level. But life has been doing it for 3.8 billion years. So this is Lodin's proposal. So this is the normal position of these protons. They're either attached to one or another atom in the DNA basis, and if they tunnel, they've got to two protons have got to tunnel in order to retain the charge balance. So what you get is like this. Whoops. You can see that you're getting a paired transport of protons across the double helix. So that's what he proposed, and when they get into the wrong position, the protons can now pair with the wrong base to cause a mutation. And that's shown here, where thymine would normally pair with adenine when the proton is in the right position, but in the rare amino position, as it's called, thymine will pair with cytosine. So it's adenine will pair with cytosine. So it will cause a mutation if that happens. So that was proposed to be a mechanism for mutation, but it's never been proven really. We've been trying to see if we could prove this mechanism. We haven't done so yet, but to show you the kind of methods that we're looking at. We grow E. coli standard bacterium that we use for many genetic experiments, and measure mutants. If you like, the parental E. coli is white, the mutants are blue. So you count the blue dots. You do this experiment in ordinary water, and in water in which the hydrogen has been replaced by deuterium, and you see if there's a difference. And when we've done that, there's water. This is the mutation rate here. For ordinary water, 50% D2O and 100% D2O. As I already mentioned with the enzyme experiments that demonstrated tunneling is very sensitive to the mass of the particle tunneling. So changing hydrogen to a deuteron, proton plus neutron, doubles the mass, slows down tunneling. So we would expect that would affect mutation rates, and indeed it does in that experiment. That doesn't prove that mutation is caused by tunneling, though. The trouble with working with biology, even bacteria are extraordinarily complex, so there are other ways of explaining this result. And we need to go to a more molecular level to see if it really holds up.
Can you give a sense of what would be some alternative explanations?
Well, if, for example, the enzymes enzymes that will work to replicate DNA also have a fidelity mechanism. So they check that they've put the right DNA base in. And then there are mechanisms afterwards which are called error repair, which also check that the right information is there. If those are affected by switching from water to heavy water, then they could account for this effect. So we need to do a lot more to work out whether it really is down to tunneling. So those were the candidates for quantum biology that I've briefly discussed. Photosynthesis, enzymes, magnetoreception. I've left out smell. Smell has also been proposed to involve quantum tunneling. DNA mutations I've mentioned. Origin of life I haven't mentioned. And consciousness I haven't mentioned either. I think other people will be talking about that. Oh, smell, I'll just give you the puzzle of smell. These are molecules that... the puzzle of smell is thought to be the specificity of smell. We're very good at distinguishing different chemicals. And yet, chemicals that appear very different can smell identical. Chemicals that look very similar can smell very different. So there is a bit of a puzzle in what's called the lock and key mechanism of smell. That it doesn't seem to fit the structures of odorants. You would have thought if there is a lock, this key opens, this one would also open, but they smell very differently. You would have thought that these two very different looking structures would open different locks and give us different smells. Anyway, Luca Turin came up with a quantum mechanical theory of smell that's supposed to account for that. Again, it's rather speculative. Okay, so those are the candidates for quantum biology. Lastly, I want to point out again that life has been using quantum technologies for 3.5 billion years, and that we propose, Jim and I, in the book that we've written, that life, come back to that original question I asked, life is unique, remarkable, and wonderful because it is able to reach down into the quantum substrate of matter to do all those strange but very powerful things like quantum tunneling, quantum coherence, maybe even entanglement, to help keep us alive. And that's, we think, or at least we proposed, what makes life special. And that's it. Thank you very much.
On the Robin experiment, the Robin case, so the high-frequency magnetic field, electromagnetic field destroyed the magnetic field of the robins and support this mechanism that you were describing. Could there be also other mechanisms?
Yeah.
Like the small magnetic particle that could also be.
Yeah, I agree.
It's not a bulletproof.
No, absolutely. It's probably better than the evidence we've got for mutation at the moment, but it's certainly not bulletproof. In order to really nail it, we need to find the mechanism that picks up, that measures the magnetic field, and find out exactly how it works. The model that Thurston Ritz proposed is very theoretical. We need to nail it onto molecules and see how it works, and then perturb it in ways that would perturb the entanglement and see that it breaks it. So it's consistent with the theory, and as far as I know, there's nothing else out there that can explain those observations except that he's right. That doesn't prove the theory, but it makes it a bit harder to explain. I mean, why should the robins? Yeah, you can't. There's nothing known. You can always speculate that maybe this or this, perhaps high-frequency radio waves are picked up by neurons in the brain, that they may interfere with the signaling pathways for the compass. But it's speculation in that direction as well. So consistent, but doesn't prove.
You have this slide with the described the developmental process, and you said that the quantum effects are amplified through the developmental process to create a phenotype.
Yes.
So, but on the other hand, we have a theory of gene expression which describes described by the genotype-phenotype mapping.
Yeah.
So, what does the quantum perspective add to this classical theory of gene expression?
Yeah, well, as you say, amplification is a key aspect of what we're claiming. That events that take place at the quantum world can have impacts at the macroscopic world. For example, if a mutation happens in your DNA and that mutation has been generated by a quantum tunneling event, then that can change the colour of your eye. So there's that kind of thing. We're not proposing that the whole system remains quantum coherent, but just that it remains quantum coherent for long enough to make a difference to the cell. So one of those differences could, for example, be a mutation taking place in the cell. Another is enzyme action. Enzymes will manipulate individual particles at a quantum level, and then because they have this huge amplification, they can speed up a reaction by a factor of 10 to the 20, then that speed up of a quantum level reaction will make a macroscopic difference to the phenotype of the cell if that enzyme, for example, is required to keep it alive. The fact that it can speed up the reaction rate may be the difference between life and death. Okay?
Very nice talk.
Thank you.
One of the things that I like about quantum biology, from what little I know about it, is that you're including Planck still in the equations,
Yeah.
which gets lost completely when you get to quantum light at the conscious level or the social level. But I also like your comment about that what happens in the quantum world can have an effect at the larger world and the microscopic. So there is somehow or another the Planck effect should be washed out. Or the way I like to think about it is that we haven't yet found a way to encapsulate mass. We have nothing that measures mass. We can measure at the social level of consciousness. I wondered if you could maybe talk about that.
Consciousness and quantum mechanics is, I think, it's the most speculative area of quantum biology. There's no question that there has been a very good paper which made a good theoretical case that some aspect of how neurons work, ions traveling through ion channels in neurons, may be quantum coherent, and that may be used in thinking. But, and that's, I'm not sure how you asked about mass. I don't really know if that's, well, it's dependent on mass, coherence, but I don't know if that's important in that respect. But the key thing is that the quantum stuff has to be at a level at which it makes sense. All the systems that I talked about, the quantum mechanical phenomena were occurring within a single molecule, such as a photosystem, a single DNA molecule, or a single enzyme. That's as far as the quantum coherent stuff goes. That can then have a macroscopic consequence because life has this unique capability of amplifying from the quantum level to the macroscopic level. But it's at the quantum level that coherence and all the weird stuff stay. It's got to make the difference at the quantum level, which is then amplified to the classical level. And that's the key thing we have to bear in mind with quantum biology. We can't have coherence going all the way up to large things, bigger than a molecule or a few molecules, really. It's just not feasible.
Any other question will be addressed individually to Professor McFadden. Thank you very much.
Thank you.